My name's Alissa Bleem. I'm a staff researcher at the National Renewable Energy Lab. And today I'm going to discuss some tools that kind of nicely build off of what Adam was just talking about. These are different genome engineering strategies that we and others have developed to really look at the whole genome of different bacterial hosts and identify genes that are important for growth, tolerance, or whatever desired phenotype you may be interested in. So next slide, please. And I think this, again, builds nicely off of what Adam was saying, because there's different things that we might want to engineer for, right? So if we have a totally new organism that we know is good at making something or tolerating something, but we don't know much about its genome, we might want to annotate those genes more thoroughly. Conversely, if we have an organism that we know a lot about, so in the ABF, this is a good example of this would be Pseudomonas putida. We might know what a lot of the genes do, but we don't necessarily know how they function together or how they function in certain contexts. So this is kind of an illustration of that idea. If you're going to try and engineer a strain or learn more about it, you really need to know what the genes encode and what the function of those proteins is, right? So having a priori knowledge or automated annotation of a genome is helpful, but it can only get us so far. So in this top cartoon, I'm showing, let's say that if I know that this top gene is a member of my pathway and the bottom gene is not a member of my pathway, okay, I can go ahead and overexpress that gene that I know is important and maybe ignore the one that I know isn't. But there's loads of other genes, right? And I don't know how those are going to influence my process. And getting a better idea of how those genes function in given contexts can help me make a better optimized organism. So that's kind of the bottom part here, where a gene that's important for growth in condition A might not be important for growth in condition B. And being able to find out all of these things in high throughput can be challenging, but we have some really great tools to overcome that. So next slide, please. So we have 3 kind of genomic library tools that we can use to help assign gene function in bacteria in the ABF. The first one that Adam already touched on, and I'm not going to talk about, is CRISPR interference. So this is basically the ability to build a library where every gene in the genome is knocked down. The second one is in the middle. This is where we use transposons to functionally knock out every gene in the genome. And we call this an RBTnSeq library because we use random barcodes to map the location of each transposon insertion in the genome. And then on the right, this is our last tool where we can actually overexpress each gene in the genome. And we just do this by copy addition, either on a plasmid or using genomically integrated constructs that are added to the genome via SAGE. So next slide. I just want to talk about some different applications that we've demonstrated for these RBTnSeq libraries. This is something we've worked a lot with at NREL and across the ABF really, but these are some demonstrations that we've done here. So one thing that we've done in Pseudomonas putida is used these randomly barcoded TnSeq libraries to identify genes that are important for stress tolerance. So bacteria in industrial settings experience a lot of different stresses, including, in many cases, exposure to high levels of salt. So basically what we did here is we took this randomly barcoded Pseudomonas putida library where every gene in the genome is disrupted by a transposon, and those transposons have barcodes, right? So we can grow the strain in a rich media where the barcodes are present at some known abundance. And then we can also grow the library in a stressful condition, so this high salt condition. And you can imagine that if certain genes are important for growth in that condition, they're not going to be able to survive if they don't have that gene present, right? So the barcode abundance will shift based on the growth condition. And this can give you an idea of what the fitness of each gene is. So basically, you're just counting these barcodes to determine these gene fitness values that I'm showing in the heat map on the right. And we can use this to determine genes that are important for growth. So things with a negative fitness value are genes that were depleted from the population. That means that those genes are important for growth. And then things that have a positive fitness value actually mean that disruption of the gene was beneficial for growth. So these are kind of nice hits. We can look at PP1430. That's a gene that we didn't really know its function in Pseudomonas putida. But if you knock out that gene, you can see in the plot on the right that the strain actually grows a lot better in the presence of high salt than its parent, if that gene is knocked out. So this is a great kind of non-intuitive discovery tool for genes that might improve your strain under desired conditions. Next slide, please. In production strains, we can also use TnSeq to enhance the production of a desired product. So again, this is in Pseudomonas putida, but this is in a strain that's not the wild type. It's actually engineered to make muconic acid. So again, we can grow our strain in rich media and compare it to our desired growth condition. In this case, it's a sugar media that we use to convert toward muconic acid as the product. The plots on the right are growth in bioreactors. But basically what it's showing is that if we have our transposon insertion library in blue relative to just the parent strain, we actually get better growth and higher product titer. So this indicates that some number of genes in the population can be either enriched or depleted to kind of create an optimum gene fitness set. And by looking at these data, we are now identifying targets to identify strain engineering opportunities to make better muconic acid production strains. Next slide. So overall, the impacts of these tools, I think, could be applied really broadly. Right now, like I said, we're mostly using them in Pseudomonas putida in the ABF, but they're adaptable to basically any gram-negative bacterium and maybe gram-positives depending on the transposase applicability. So the methods, protocols, and computational analysis pipelines that we use for this are really readily translatable to other hosts. So if you have a host that you're interested in learning more about its genome annotation, we can leverage these tools pretty directly toward that. We've also done a lot of work to make sure that the experimental designs are straightforward, so we can heavily multiplex these experiments, doing up to 96 different enrichment conditions in one sequencing run. And again, the analysis code for this is open source and readily usable to scale. And finally, we can combine these libraries with other screening techniques. So we can actually take our transposon insertion library and array it out. So each colony, each strain has a different transposon insertion. And then we can screen those colonies for example, production of muconic acid. And in that way, we can learn much more better links between genotype and phenotype.